Welcome to another episode of the Life Science Solutions Podcast, where we explore the technical, operational, and regulatory challenges shaping the future of the life sciences industry. I'm your host, Chris Adkins. Today, we're diving into a topic that sits at the intersection of formulation science, analytics, and manufacturing strategy, and that's particles in biologics and their potential for impact on technology transfer. Particles and aggregates are more than just a test result at the end of development or an issue that pops up in manufacturing. They can reveal deep insights about formulation stability, process robustness, and potential risks when moving a product from development to manufacturing or between sites. As biologics become more and more complex, higher concentrations, new modalities, advanced delivery systems, understanding particle formation and characterization becomes even more critical. To help us unpack this topic, I'm joined today by Tim Menzen, Chief Technology Officer at Coriolis Pharma, a science-driven CRO based in Munich and with a location in North Carolina that focuses on formulation development, lyophilization, and advanced analytics for biopharmaceuticals. Tim is a pharmacist by training and earned his PhD at LMU Munich, where his research focused on temperature-induced unfolding and aggregation of monoclonal antibodies. Since joining Coriolis in 2014, he has led biologics formulation projects, implemented novel technologies, and now oversees internal research, as well as collaborations with industry and academia. Tim has also been deeply involved in advancing particle characterization approaches and digital tools that help scientists better understand particle populations across the development lifecycle. Tim, welcome to the show.
Thank you for having me, Chris.
Yeah. I'm really looking forward to this conversation. Before we deep dive in particles, and I've got my battle scars from that. But to start, can you tell us a little bit about your role as CTO at Coriolis and how you typically explain the work that your team does to either development or manufacturing leaders who might not have as much familiarity with you all?
Yeah, sure. Thanks. So in order to understand the role as CTO at Coriolis, you have to understand Coriolis in general, right? So we are a purely service provider, analytical services, but our core expertise is formulation development of biopharmaceuticals. Mm-hmm. So that's where our operational teams focus on, helping our clients to deliver life-saving medicines to patients. So that's what we're here for. Mm-hmm. And in order to maintain Coriolis as an innovative service provider and solution provider, so we really want to enable our clients. We have a unit that's called Science and Technology. So that's a unit that I lead together with Andrea Have, our CSO, and we there basically try to solve the problems that we see in our daily labs, right? Just an example, polysorbate degradation. So that's something that we have developed methods for characterization of polysorbate degradation, but also somehow prepare us for the molecules of tomorrow. So when a client approaches us tomorrow with a new modality, that we have an understanding what we should do about that. So basically that's in a nutshell the summary of science and technology, so that we are innovative and that we have good solutions. And in my personal responsibility, I'm responsible for our internal research projects. So these can be things that we just run strategically at Coriolis, but also many collaborations that we have academia and industry partners. And another team is focusing on digitalizing our labs and data science. So that's in a nutshell.
Okay.
Responsibility.
Yeah, really exciting. And you know, it's really good, and we're going to talk a little bit more about your background, but it's really good to get to have conversations with folks who are really on the cutting edge, and that's certainly where you guys are on the formulation development. You're talking about new, whether it's new molecular structures or new ways of doing formulation and things like that are so critical. I mean, it parallels so much with what we see on the device innovation side, right? With the work that we do in that space. So it's interesting. I'm looking forward to chatting. A little bit, into your background. Mm-hmm. It includes, and I made reference to it from your PhD program, research on antibody unfolding and aggregation. Mm-hmm. How did that early work shape how you think about particles and overall developability in biologics today? Yeah,
it's interesting. I think it's a rare case that, like me, I could apply the PhD know-how that I gained during that time immediately at Coriolis. So I think that's a rare case.
Yeah.
So I was studying during my PhD a lot of thermal unfolding. And then usually this leads to protein aggregation. And you expose hydrophobic patches, the molecule starts to aggregate. So I learned a lot about basically the impact and also the links between conformational stability and then colloidal stability, so protein interactions. And that's very exciting. And also testing the impact of formulation. So that basically paved the way for then formulation development projects at Coriolis then after my PhD.
Yeah, that is so rare to have PhD research be so applicable to what you immediately go into in the industry. So that's very fortuitous for both you and for Coriolis, right? I mean, because you have a PhD in something, that means you're a global expert in that specific area of science, right? Or at least that's how I always thought about it. So, so from your vantage point, right, at a science-driven CRO and being an expert in your field, what trends are you seeing in the types of development or in the manufacturing challenges that companies are bringing to you, right? And how have you seen that change over the last decade?
Yes. So I think when I was still, let's say 15 years ago doing my PhD, the engineering of molecules, let's say antibodies, was not that advanced. And you still had, let's say, these bad performing molecules exposing high viscosity, aggregating like hell. So I think that's something that we have learned somehow to engineer in a better way. So I think there's a lot of improvements in protein stability in the extrinsic protein nature. So, and this also brought in a way like a paradigm shift in how we handle formulation for those molecules, right? So for an antibody, you would probably don't start from the scratch. You have learned a lot about how antibodies behave. And then basically it's more about formulatability assessment. So is this molecule a good behaving one, or is it basically one of the challenging ones?
Yeah.
And then if you don't see really like risks, you could probably just go into stability studies with a limited set of formulations, and you skip a lot of the pre-formulation work that we have done in the past. And that's a little bit of a paradigm shift, at least for the antibodies. I think for the traditional biologics or any other protein, we have not that understanding, and we have to really basically do a good formulation work with all the screenings and surfactant screenings and all that.
Yeah. The more the cruder way of approaching it, right? Yeah. And doing, maybe getting as far as doing some good, DOE work and whatnot, right? Yeah. But yeah, I mean, it's really interesting though how we've moved and I mean some of this, kind of Hawkins towards as we think about some in silico modeling and things like that, and some really good, really legitimate uses for artificial intelligence and machine learning and some of the other work that you guys are doing kind of adjacent within Coriolis. But it has shifted that side of the space so much.
Yes,
right.
We are doing a lot of simulations as well now.
Yeah. And it's really, I don't know, to me, it's really intriguing because it has shifted so much so fast, and not only because of the technology, but I mean, we've moved from, we just have kind of a database of, excipient and formulation profiles that we saw that it worked before, so we're going to try it again. And it's our maturity of the understanding of the system has improved so much.
Yes.
Right.
And it's still a matter of risk. Right. Yeah. So how much risk are you willing to take?
That you're going to start to see aggregation at month 13,
for instance,
right?
Particle formation.
Exactly. Right. And then how does that risk impact how many formulations you try, what packaging components you try, right? Yes. Do you actually risk trying, I come from elastomer manufacturing, do you actually risk trying uncoated to see if that can work because of long-term supply availability and economic realities, right? Not to mention machinability challenges if you're actually going to make 1 million or millions of doses a year. So
yeah, I think you make a good point. There's a lot of things to consider beyond formulation, or let's say beyond the stability of the drug. Yeah, that should be considered in the target product profile.
Yeah, absolutely. Right. There's some really great packaging technology that is prohibitively expensive in the industry still. Yeah. And maybe that's just my vaccines background coming into play. So when you hear the phrase technology transfer for a biologic product, right? And especially, we live in a world now where even if at phase 1 by some, you're operating at small enough long-term commercial volumes that you could reasonably do phase 1 at a commercial scale, right? Those products do exist. They're few and far between, but no matter what, you're going to have technology transfers along the way. But when you think about technology transfer, what particle-related risks immediately come to mind?
Yeah, I think I would say pumps.
Okay.
And I would cite Professor John Carpenter here, because I think he had said that pumps are particle generating machines. And I think that's probably the thing that comes to my mind first.
Yeah. Not what our friends at Watson Marlowe necessarily want to hear, but so what makes you say that? Just what they do to the protein as you're going through the peristaltic process, right?
Yeah. So first of all, I think there are different types of pumps.
Of course.
And then I think then there are a lot of stress factors associated with the process of pumping, which could be a problem for protein stability and then particularly particle formation.
Yeah. So and that's a good point, right? And we never have the luxury of doing a exactly like for like tech transfer, right? That does not exist. That would be great, right? But even within big pharma to the same within the big pharma, from their pilot to their product, it's not exactly like for like. And some of that's process scale realities, right? That you don't have the luxury of just putting some C-flex tubing in a peristaltic pump anymore, and you have to go to a more advanced pump technology. So,
or having like a good downscale model.
Yeah, exactly, right. And I mean, there's tons of advantages there, but there's significant cost in getting it validated properly, right?
Yeah.
But obviously I come from a TD background, so that would have been fantastic to have. So many teams think about particles primarily in the context of investigations when issues arise, right? The first half a decade of my career, this was entirely my exposure, and then it's a fire drill. From a scientific perspective, though, what can particle populations actually tell us about formulation stability or process stress at the development phase of a program?
Yeah, I think what you're alluding to is basically the red lights that flashing when you see visible particles. So I think that remains always like the biggest signals of having a problem. But I think when you characterize particles in general, and you also look into subvisible particles or submicron particles, you can see that there are process-related things and product-related origins of particles. And if you get a good process, but also product characterization, you understand the liabilities of your product, and you have a good understanding of what actually is causing these particles before they pop up as these visible particles, and then when you really the problem is already like advanced. Yeah,
correct. There's some good legwork to do, right? That if you understand some of that subvisible, and then you end up in a space where it becomes visible, right? Your issue has been there the entire time. In actuality, you've now just crossed the regulatory line where it's not okay anymore, at least in most cases. There's some strange point of use filtration things that are allowed on subsets of products. And if you don't work in those products, what I just said sounds insane, but it's true. But you can have that learning, and you can actually understand your process, right?
Yeah, I think there's never a particle-free product. So even if you filter with a sterile filter 0.2 micron, there'll still be submicron particles. So the particles are always there.
Yeah, right. Just because you don't see them doesn't mean they don't exist. So, Coriolis really emphasizes broad particle characterization using this, and I'm going to let you define it because we're way over my scientific skis, but orthogonal analytical techniques, right? And so one, define for us what that broader kind of analytical toolbox is, and then how does having that capability and the knowledge that you get from it, how does it change the questions that teams can answer as they're doing process development and ultimately tech transfer? Yeah. So I think most of the audience will be familiar with the pharmacopoeia methods, test for visible particles, and then the subvisible particles with light obscuration, membrane microscopy, and so on. So there the focus is really just on sizing and counting particles. And this is okay for QC, right? So that's where the pharmacopoeia methods are intended for. And I think during development, you really want to look deeply into your product and characterize all the particles that are there, a completely whole size range. As we talked about, that's basically from the dimers to the visible particles. So, and then I think a lot of information is obtained from the methods that add you some additional insight. So not just sizing and counting, but maybe also get you an idea about what type of particles
particle you're looking at. So being it morphology-based, is it looking like a sphere? So it might be silicon oil versus like a fluffy protein aggregate from looking at a microscope image. Yeah. And so that's what we like to do. So doing extended characterization and then getting as much information out of the product, and then enables us, coming back to the second part of your question, doing the right design decisions, being it formulation, but also being the packaging material, silicon oil coming from the containers and so on.
Yep.
So,
Yeah, absolutely. No, that's a very good point. So before we keep going, I do want to just give a quick plug to the folks who are listening. If you're enjoying the episode, give us a like on your player of choice. Consider subscribing wherever you're getting this podcast from, and more importantly, share it with someone in your network who maybe has had to sit with you and write a nasty particle deviation, and you wish you understood why the heck it was happening. But it really helps more folks discover the show. So, Tim, can you share an example at a high level where deeper particle characterization helped reveal an issue in development or in tech transfer that traditional testing that you just talked about would have just totally missed? Yeah.
So immediately to my mind comes an example where we supported an investigation when there was a particle that we thought may come from the stopper, contain fluorine. And looking deeper into that, we could demonstrate that actually not coming from the stopper, but from a different part of the process. So I can't go into detail here, but
sure.
That was, for instance, like something that sticks.
Wow. I have a lot of former colleagues in the elastomer space that love what you just said. They wish that could always be true, because that's also not a particle-free process, but certainly one where we reduce as much as we can. So kind of in that vein, you've been really involved in a lot of work around this idea of particle fingerprints, right? And we can all kind of guess at what that means, and having some AI-enabled analytics. What exactly is a particle fingerprint, right? And then why is it so useful when we compare batches or manufacturing processes, right? And we leverage that for some comparability work.
Yeah, I mean, getting images of particles from these systems like flow imaging microscopy is very tempting, right? So to use, let's say, AI to do computer vision tasks on these particle images. And I think the first obvious thing is to do a classifier. That you train a machine learning model by using a data set that you have curated, and that you know what is the nature of the particle in the training data set, and then you get a per image or per particle classification. So is it like a silicon oil droplet, or is it a protein aggregate? And the idea of this fingerprinting approach, and that was basically published some time ago by Chris Calderon and colleagues, and it's also commercial software available, Century Sciences, I'm not sure if I can drop the name here. So that allows to do more like a dimension reduction of the features of these particles, and it ends up in a two-dimensional space, and every particle is then just basically populating one point in this two-dimensional space, and you get a density distribution, and that looks like a fingerprint, and that helps you to see if a whole distribution of particles is similar or not. Right, and if you basically then compare two processes and you build a model and compare it, if the particles morphology looks similar, these fingerprints would overlap.
Yeah.
If the morphology is different, suggesting maybe a different mechanism of particle formation, you have the fingerprints in different regions.
Okay, and so do you see folks leveraging these advanced analytics as they're doing process transfer, right?
Yeah.
And then they're comparing not just what is maybe a traditional, obviously they're gonna be meeting their QTPP and release testing and all that kind of stuff, but doing this as kind of a next level of analytical or comparison at the end of the day?
Yeah, I mean, this tool is perfect for doing this A versus B process characterization, right? Because you have a hypothesis, either it's similar or not, and you get this clear answer. But I think it's still basically very early and requires still a lot of understanding and more advanced understanding if also the morphology has a meaningful impact, right? Yeah. So I think there is still some way to go, but I think we would miss we would miss information if we don't include the morphological information. So sure, size, number of particles, that's established.
Yeah.
But I think there's also information in the morphology.
Yeah. I mean, because immediately my mind goes to, okay, well now you have learned new information, right? You can't unlearn it.
True.
Right. So from and I've come from GMP operations. There's a regulatory impact. So, I mean, I'm curious, like, we want to understand our processes better, right? Broadly as an industry, we are a science forward industry. I say that as an engineer, but we are science forward in what we do and in how things work. So there's additional data to glean here, but we just need to learn it in the right way and then approach the regulators with it in the right way, right?
Yeah, let me clarify. Probably it's I think the technology works. So if the images are different, you would get the information. But the question is this relevant or not?
Correct. Is it bad different or is it just different?
Is it just different?
Right? Yeah. I mean, because if your process isn't picky about the method of liquid transfer, what type of pump you use, right? Then it may not matter, right? Or the same on the fill finish side. If you would not normally change, but if you did, vent tube displacement versus vacuum stoppering, it may actually be that either one is okay for your product because you don't need a particularly small bubble point, right? And it's just you have two different manufacturing lines and they can be different, maybe because you have two different CDMO partners or you have internal, external, whatever it might be, right? So not all different is impactful different.
Correct.
And this is where you get back to that. How do you build this database of knowledge from development so that you actually understand what's going on in your formulation and what will drive us to an issue?
Yes.
Right.
And what are really like biological consequences.
Yeah, exactly.
Ultimately.
Yeah. No, that's good. So, as you look ahead, where do you see these advances in particle analytics, digital tools, AI and some of these analytical techniques? How do you think this is going to change the way that companies approach formulation development and ultimately, process development into tech transfer? I mean, we changed quite a bit over the last 15 years. We talked about that at the beginning of our time together. What do you think is going to be the next horizon of change over the next 5, 10, maybe 15 years?
Yeah, I think there's a lot of uptake in the domain of simulations. So what we refer to in silico, so running experiments in the computer. I think that will really bring us a lot in forward in the future. And looking into the topic of today, also particles, I think there is this interest in having more information. So instruments that provide another dimension of information, being it fluorescence or being it refractive index or something else that adds to your traditional information about particle size and counts. That's what people look for. Miniaturization, so needing less volume but still getting statistically relevant numbers from that analysis. So that will help to speed up development, save material. I think that's what everybody's looking into. Yeah.
No one enjoys at phase one manufacturing sampling out their entire production batch that it took their team 30-ish days to manufacture and then another 2 months to release or maybe longer. So, well, excellent. Tim, really enjoyed having you on. Appreciate you joining the podcast, sharing your perspective and all your scientific expertise, right? Particles are often treated as a late stage compliance issue, but this conversation obviously highlights how much insight we can glean throughout the development process, and how it can lead us into a better position for technology transfer at the end of the day if we have genuinely followed all of our QBD principles, right, and actually done good development. So, if you're curious to learn more about Coriolis Pharma, check out their website. If you're curious to learn more about Kymanox, check out our website. Thanks everyone as always for listening to the Life Science Solutions podcast. If you've enjoyed it, please subscribe, share it with a colleague. And until next time, thanks for listening. Thanks for tuning in to Life Science Solutions, presented by Kymanox and produced by Walk West. For more insights like this, subscribe and follow Life Science Solutions wherever you get your podcast. We'll see you next time as we continue exploring what it takes to bring life science innovation to life, because patients deserve better.